Our next couple of chapters that we're going to be looking at are energy chapters. They have to do with energy conversions, whether it's Chapter 6 here, Cellular Respiration, dealing with how we get energy from the food that we eat, or our next Chapter 7, dealing with how plants are generating usable energy through photosynthesis. And obviously, energy is an essential component for life. We all, as living things, need to have our energy source. And energy is linked to almost everything we can think about with living organisms, whether it's growth, transport of chemicals in and out of cells, building chemicals like proteins and DNA and nucleic acids, cell movement, or even reproduction are all certainly linked to energy. And while all chemicals contain some energy, most of the energy in chemicals is not really in a usable form. It's not in a form that cells can utilize right away for these different activities. And so Chapter 6 is dealing with the process of cellular respiration. And cellular respiration is a very complex metabolic pathway. It's a long chemical pathway of lots of chemical reactions all hooked together in these long chains. And the idea behind cell respiration is it is going to provide energy in a usable form. So it's all about energy conversion and generating usable energy. Now, cellular respiration is a pretty complex metabolic pathway, over 30 some odd different chemical reactions all needed to get this energy converted into a usable form. We're very much going to take an overview approach. We're going to look at some of the major stages. We're especially going to be interested in the energy. Where's the energy at? Where is it going? What form does it end up in? And so on. But we're certainly not going to get into the level of detail that the book gets into. And again, lots of enzymes required, lots of different chemical reactions. But the whole take-home message of cell respiration is all about energy conversion. Getting potential energy, which is stored up in the chemical bonds in macromolecules. In particular, the chemical bonds that hold carbons together contain lots of energy. And so whether it's looking at sugars or fats or proteins, in cell respiration we're going to convert some of that potential energy into usable energy, which, remember, for cells, usable energy is ATP, adenosine triphosphates. And so the whole point here is to generate lots of ATPs for the cells to use. And this process, this pathway, is going to be tied directly to the mitochondria, our powerhouse of the cell. This is where most of the metabolic chemical reactions are going to occur. And some organisms undergo cell respiration without oxygen. Others, like our cells, require oxygen. And we're going to focus on the oxygen requirement first. That's referred to as aerobic respiration. So with or oxygen requiring is aerobic respiration. And here's our summary chemical reaction of respiration. Now this is a summary. This is not one big chemical reaction, but again, what's going in the pathway and what's coming out. And notice going in the pathway, our most common source of energy is glucose or sugar. We do require oxygen, and then through this pathway we're going to break apart that glucose into carbon dioxides. So we're going to break all the carbons apart. We do produce a little bit of water as waste product, but most importantly, that energy that starts out in the covalent bonds in the glucose is going to be converted, at least some of it, into ATP form, and that represents usable energy for our cell. And breathing and cell respiration are certainly linked together. We think of breathing, we have to breathe in oxygen as our major source of air, and we're breathing out carbon dioxide as our waste product. And that is directly linked then to cell respiration, what's going on at that chemical metabolic level, because oxygen is going to be required for us to get energy into ATP form, to get that energy into usable form, and likewise the carbon dioxide waste product from cell respiration is going to be released as gas, and that's going to be breathed out. And so certainly a direct link here between breathing and our cell respiration process. This is also the reason why if you're not getting oxygen into your system, you're not going to survive very long, because if you run out of ATPs in your cells are going to shut down and start dying off within a matter of just a few minutes without that oxygen. And before we get into the actual metabolic pathway of respiration, I do want to mention a couple issues that we're going to be key here in understanding how respiration works. And some of this certainly is going to get back into some of the chemistry that we've talked about. And I know that that's kind of off-putting for some people. It's really not your favorite thing to talk about. But I just do want to go over this and make sure we understand it and then link it back into respiration. And one of the ways that cells can utilize energy and pass on energy from one chemical to another or convert energy from one form to another is by utilizing electrons. And electrons, remember, are small, they're negative, and they're very high energy. And because they're small and high energy, passing in electrons from one chemical to another is an easy way for cells to pass energy from one chemical to another. And when electrons are shuffled around from one chemical to another, that's called a redox reaction. And redox is short for reduction and oxidation. Okay? And so in these redox reactions, again, one or more electrons are going to be moved from one chemical to another. And the two components there, reduction and oxidation. Oxidation is when a chemical loses an electron. And so a chemical undergoes oxidation if it gives away an electron. Likewise, a chemical is reduced if it gains an electron. And so these reduction and oxidation reactions always go hand in hand. One chemical loses the electron, the second one gains that particular electron, and hence that's why we call that a redox reaction, because they're always going to coincide together. So for example, if we look at a picture here on top, we see a chemical being oxidated. It's going to lose an electron, and then in turn, at the bottom there, that electron is going to be passed on to the next chemical. In this case, it's a chemical called NADH. That NADH is going to gain an electron and become reduced. And so we're going to see this idea of oxidation and reduction playing a real central role in how cell respiration works. So if we relate then these redox reactions and energy issues concerning these electrons. So oftentimes what we see in cells are a whole series of chemicals, usually proteins, and electrons are going to be passed from one chemical to the next, to the next, and the next, and so on. And so there may be anywhere from maybe 3 or 4 to 8 or 10 proteins that are going to pass this electron from one to the next. And every time that that electron gets passed from one chemical to another, it's going to lose a little bit of its potential energy. So its potential energy is always going down. And this series of chemicals that are going to pass the electron along are referred to as an electron transport chain. This just chemical chain reaction of passing this electron down. And again, with each step in the chain, that electron is losing a little bit of usable energy. Now that energy that the electron is losing isn't going to be completely lost to the cell, and in fact the cell is going to harness some of that energy. So as that energy is lost from the electron, the cell is going to try to harness it, and that energy is going to be used to generate some of the ATPs, some of our usable energy. And here's just a picture from your book showing that. NADH gets oxidized, meaning it's going to lose some electrons. Those electrons then get passed from one protein to another down this chain of chemicals. So that's our electron transport chain. At each step it loses a little bit of energy, and that energy is used to harness ATP. Now eventually oxygen is the chemical that's going to bind up the electrons, and that's why oxygen is so important to our cells. It binds the electrons and it runs these electron transport chains. Now the analogy I always think about when I think of these electron transport chains are slinkies. And so I have my little slinky picture here. When you think about slinky going down the stairs, each time it takes a step down, think of that as an electron stepping down the electron transport chain. It's getting lower in energy with each step, just like a slinky gets lower and lower down the stairs. So with cell respiration, it's going to involve many chemical pathways, but certainly the electron transport chain and this idea of passing energy through electrons is going to be an integral part of how we're going to take energy stored up in those carbon-carbon bonds and things like sugars and fats and so on, and convert that energy into ATP, which again, your cells constantly need ATP. Every minute, your cell, a single cell is probably using millions of ATPs worth of energy and has to regenerate those ATPs constantly again to stay alive. Now, if we look at aerobic respiration with oxygen, our method of respiration for humans and other animals, any organism that requires oxygen, plants as well, there are 3 main chemical stages or pathways to get us from potential energy to ATP. And those 3 stages are glycolysis, the citric acid cycle, or sometimes it's referred to as the Krebs cycle, and then oxidative phosphorylation. Now, other organisms can do this without oxygen. That's called anaerobic respiration, when there's no oxygen present or no oxygen needed. For example, some yeast and some bacteria can get ATP energy from sugars without oxygen. And so what we're going to do with this chapter is we're going to break down aerobic respiration, just go through those 3 main stages of getting energy into usable form, and then near the end we'll take a look at what happens in organisms that don't use oxygen, that are anaerobes. With oxygen present then, our first series of chemical reactions is called glycolysis. And again, this is a pathway in itself. This is several chemical reactions all linked together, and this is actually occurring out in the cytoplasm. And the point of glycolysis in this process is to start to break down the sugar. We're going to take glucose and we're going to start breaking those carbon bonds apart. And as those carbon bonds are broken apart, that's going to release energy. Now, a small amount of that energy released as we break the glucose apart is going to end up being usable energy. And usable energy is always ATPs. But some of the energy that's released in that breakdown of the sugar is going to end up in a different form. And the form of that energy is a chemical called NADH. And NADH is a small chemical. It's similar to ATP in that it's high energy, but what's different about NADH is it's not in a usable form yet. Your cell's going to have to process this energy, this chemical, to get the energy out. And so in glycolysis, an NAD, which is nicotinamide adenine dihydrogenase, we'll just call it NADA for short, is reduced, meaning it's going to gain electrons during this process. Once it has those electrons, it's got more energy now, because remember, electrons we're going to associate with energy, and so the cell is going to process those NADHs later to convert that energy into ATP form. And so here's a nice summary reaction of glycolysis. Again, we're going to take glucose, we're basically going to break it in half. When we do that, we're going to generate 2-pyruvate. Each pyruvate is a 3-carbon chemical, so each of the little balls here in the diagram represents a carbon. Breaking those bonds is going to that chemical bond is going to release a little bit of energy. We'll get a couple ATPs of usable energy, and we'll get a couple of this NADH of this coenzyme that the cell's going to have to process a little bit later. And so at the end of glycolysis, we've got pyruvate. We're going to send that along to our next stage, which is the citric acid cycle. Once that pyruvate is generated from glycolysis, then it's going to be passed into the mitochondria. And the mitochondria is where the rest of our chemical pathways are going to occur here with cell respiration. Remember, the mitochondria, that's our powerhouse of the cell. It's going to generate most of these ATPs that come in your cell. And the main take-home message here with the citric acid cycle is that's where the sugar, those pyruvates that were just generated from that sugar, are going to be broken down completely. And so all the carbons are going to be broken apart from each other. So that original glucose, by the time we're done with the citric acid cycle, will be completely dismantled. All 6 of those carbons will be pulled apart. And all those carbons are going to end up being converted into carbon dioxide. And so this is our main waste product, carbon dioxide. This is where the carbon dioxide comes from that we breathe out in our air every day. And it's kind of a weird thing to think about, but when we breathe in air and breathe out carbon dioxide, the carbon dioxide we breathe out didn't come from the air that we were breathing in. The carbons that we breathe out in carbon dioxide actually are coming from the food that we ate. The carbons from the sugars and fats that our cells metabolize get converted into carbon dioxide, and that's our way to get rid of it, is to breathe it out every day. So kind of an odd sort of thing to think about. Now, as we break apart our sugar here in the citric acid cycle, we're going to get a small amount of usable energy again. So we'll get a little bit of ATP, but most of the energy, by far the bulk of it, is going to end up in the form of those coenzymes. Not only NADH, but we'll see a similar chemical called FADH2, which is another small high-energy chemical. And so these coenzymes are going to be reduced during this process. They're going to gain some electrons, and again, we're going to have to continue to process those eventually to get them into usable energy form. So let's take a look at the citric acid cycle. So here's our summary of the citric acid cycle, and again, we're not going to get caught up in all the intermediates and all the names of the chemicals in these processes, not because they're hard, but because we're just not going to take the time. We're going to spend our time learning other things this semester. We could spend weeks alone, certainly on the cell respiration here. But if we look at the citric acid cycle, the carbons are going to be broken apart and released as carbon dioxide, so we can see our waste products there, our carbon dioxides. We're going to generate a little bit of usable energy, an ATP, and then we're going to generate lots of NADHs, 3 of them in particular, and 1 FADH2, which is where the bulk of the energy is going to be contained. The original sugar contained 6 carbons that got broken into 2 pieces, or 2 halves of pyruvate that we saw in glycolysis. And so this citric acid cycle is going to turn twice for each sugar, because each half of the sugar is going to be processed here. And so in reality, for every sugar we start with, we're going to get 2 ATPs, 6 NADHs, and 2 FADH2s, so a pretty substantial amount of energy. And in turn, we're going to end up generating 6 carbon dioxides as waste product. Our third and final stage of aerobic respiration is oxidative phosphorylation. Now, once the citric acid cycle has been complete, there is no more sugar left. The sugar, glucose, is completely gone. Now, this set of reactions will occur in the mitochondria as well. In particular, they'll be associated with the inside mitochondrial membrane. And what's going to happen in this stage is we have to process those coenzymes. So all those NADHs from glycolysis, the NADHs and FADH2s from the citric acid cycle are all going to be shipped inside the very center of the mitochondria. And in oxidative phosphorylation, the mitochondria is going to process those coenzymes and generate ATPs from that energy. This is by far the high energy step. On average, we get about 38 ATPs worth of energy from a glucose molecule. 34 of the 38 come from this last step, this oxidative phosphorylation. And so this is certainly the step that generates most of the usable energy. This is also the step that requires oxygen. And so oxygen is going to help drive or run this stage or these metabolic reactions. And we can break up oxidative phosphorylation into 2 major groups of chemical reactions. One group is electron transport, which we already talked a little bit about, and then the second is a process called chemiosmosis. So let's take a look at these two processes and how we're going to convert that energy from coenzyme form into ATP form. And we mentioned the electron transport chain earlier as being an important aspect of how energy is going to be passed and transferred here. And so the electron transport chain is going to represent a whole series of these redox reactions, these electrons being passed from one chemical to the next and then to the next one and so on. And the electrons in these redox reactions, the energy is going to come from these coenzymes, these NADHs and FADH2s. And so these coenzymes are going to take their electrons and they're going to become oxidized. They're going to give away their electrons, and when they do that, they're going to reduce the first chemical or the first protein in the electron transport chain. And then that electron is going to be passed right down the chain. Now eventually that electron is going to be bound up by oxygen, and this is why oxygen is so important, because oxygen is very electron hungry. Electrons are very attracted to oxygen nucleuses, and so the oxygen will pull the electron off the chain. In doing so, that oxygen gets converted to water. That's where the water comes from in our process. But more importantly, every time oxygen pulls an electron off the chain, that pulls all the electrons down the chain, and that frees up space then for another electron to go start the chain. And so it kind of keeps this chain of electrons running as oxygens bind and pull the electrons off the end of the chain. Now remember, as electrons pass down this chain, every step they take, think of the slinky going down the stairs, they're losing a little bit of energy. That energy that they're losing is going to be used to work a couple of active transport proteins. And remember, transport proteins help move chemicals across membranes. In active transport, we said that requires energy, and that energy is going to come from these electrons. And so these proteins are going to pump hydrogen ions, or H+ ions, across this mitochondrial membrane. And all the energy from these NADHs and FADH2s are going to run several of these pumps, pumping lots of H+ across the membrane. And what this does is creates a huge concentration gradient, because most of these H+ ions are going to end up on one side of the membrane, and we're going to get very few on the inside because we're pumping them out, and ions cannot cross the membrane, and so they're going to be stuck on one side. And this is going to be critical for how our next stage works. Our second portion of oxidative phosphorylation is chemiosmosis, and this is going to be the portion that's actually going to generate the ATPs, generate our usable energy. And we talked about osmosis in our transport in Chapter 5 as being the diffusion of water across a membrane. Well, here with chemiosmosis, it's the diffusion of a chemical, any other chemical, across the membrane. And so when we finish the electron transport chain, we set up this concentration gradient of H+ ions, lots of hydrogen ions on one side of the membrane, very few on the other. And remember, our natural flow of chemicals is to spread out, is to undergo diffusion and move from high to low concentrations. And so chemicals normally are going to move with that concentration gradient. So there's a huge natural tendency for these H+ to come rushing back through, to come rushing back in the membrane and spread out. The problem, of course, is the membrane is a barrier. Because they have a strong positive charge, they cannot fit through the phospholipid or the fatty portion of the membrane. And so the membrane's impermeable. And so again, we end up with all these H+ on one side, none on the other. And the way that cells are going to generate energy then is through this process of chemiosmosis, because this concentration gradient represents potential energy stored up. All these H+ with a tendency to rush in and nowhere way for them to rush in. And a good analogy to this is think of a dam holding back water. Okay? When we have a dam holding back water, there's lots of water on one side of the dam, not very much water on the other. And the way we can use that then to generate electricity is if we open the gates at the bottom of the dam, the water is going to come rushing through with a lot of force, and we can harness that energy of that water rushing through with turbines that are going to allow us then to generate electricity and store up that energy. Well, the same thing's going to happen here at the cellular level, a much smaller level, but instead of a dam holding back water, we've got a membrane holding back H+. And if we poke a hole in that membrane or allow those H+ to come through, they're going to come rushing through with a lot of force, and cells are going to be able to harness that energy in chemiosmosis. So if we take a look at chemiosmosis, the hole in the dam, or the only place that these H+ can come rushing back through that membrane, is through a particular protein called ATP synthase. So ATP synthase does represent the hole in the dam or the floodgate. That's where the H+ can come rushing through. And ATP synthase is a couple of different proteins that basically create a turbine. And so as H pluses come rushing through, they can only fit single file through this protein, and they're going to rush through with a lot of force and energy because of this strong concentration gradient. And as they do, they turn the little protein turbine, and that turning of that turbine is going to generate energy that can be stored up as ATP. So again, very analogous to our dam. And so if we put our electron transport and chemiosmosis together, here's a nice summary figure of the whole thing. NADH, if we start on the left here, is going to donate its electron to our first protein in our electron transport chain. And then that electron is passed from one protein to another down the chain. As it does so, the energy that that electron loses is going to be used to work, in this case with NADH, it's going to work 3 pumps. And each of those pumps is going to pump out a hydrogen ion, an H+. Now eventually that electron gets passed on to oxygen, and that generates a molecule of water. Now FADH2 is a coenzyme with a little bit less energy, and so it only has enough energy to run 2 of the pumps. And so you can see it enters the chain a little bit later. Its electron will run 2 pumps and pump 2 H pluses out. So we create this huge gradient with lots of hydrogen ions on one side, very few on the other. And the only place they can come rushing in is through this ATP synthase protein. As they do, they generate a turbine, which is going to take energy and store it up into ATP form. So for every H+ that comes rushing through, that provides enough energy for 1 molecule of ATP. And so NADH, its electron has enough energy to generate 3 ATPs because it can pump 3 H+ out. Likewise, FADH2 has enough energy to generate 2 ATPs since it runs only 2 of those hydrogen pumps. And I know certainly that the chemistry is very heavy duty on some of this aerobic respiration. In fact, if you look at some of the pictures in your book, they go through all the intermediates, and we're simply not going to take time to do that. But if we take a look at an overview here of aerobic respiration, just looking at the products and the reactants, what goes in, what comes out, and then, again, where is the energy? So our first stage of reactions are glycolysis. In glycolysis, glucose is going to be converted into 2 pyruvates. Those 2 3-carbon chemicals, that's going to release some energy. A little bit is usable ATP, some of it in the form of that NADH, our high energy coenzyme. That pyruvate then is going to enter the Krebs cycle, or the citric acid cycle. That pyruvate is going to be broken apart. Each carbon is broken off so that eventually all the carbons are going to be in the form of carbon dioxide, and we're going to lose that as our waste product. That's going to release a huge amount of energy, some of it in ATP form, but most of it in those coenzymes, the NADHs and now the FADH2s. And again, by the end of the citric acid cycle, our sugar that we started with is completely gone. It's all been converted into carbon dioxide. Our last stage then is our oxidative phosphorylation. We're going to take those coenzymes high in energy from our first 2 stages. We require oxygen to make this happen. It's going to generate water as a waste product. And in turn, by that electron transport and chemiosmosis, much of that energy in those coenzymes is going to end up in ATP form. So if we look at our summary chemical reaction, the glucose is used during glycolysis. Our other reactant was oxygen. That's used during oxidative phosphorylation. Our 2 waste products are carbon dioxide from the citric acid cycle, water from oxidative phosphorylation, and ATPs come from all 3 stages, but by far most of the ATPs are coming from our last stage there, that oxidative phosphorylation. If we take a look at that energy, here's just a chart showing where the different reactions are occurring: glycolysis in the cytoplasm, the Krebs or citric acid cycle, and oxidative phosphorylation occurring inside the mitochondria. We can see glycolysis generates about 2 ATPs per sugar. The citric acid cycle, another 2 ATPs of usable energy, and then about 34 ATPs of energy from oxidative phosphorylation. And these numbers certainly vary greatly between different cell types and the environmental conditions around a cell. But in general, we get about 38 ATPs of energy for sugar, which is about 60% of the energy. About 40% of the energy in glucose is simply going to be lost, usually as heat, as we undergo these metabolic chemical reactions. Other organisms can generate ATPs from sugars without oxygen. And again, that's anaerobic respiration with no oxygen. Sometimes that's referred to as fermentation. So if we hear fermentation or alcohol fermentation, that's what we're referring to as organisms generating ATPs with no oxygen. This is a much simpler process. It's also a lot less efficient. We're not going to see as much energy here, but there are basically 2 stages. The first stage is the same, and that's glycolysis. And so it doesn't matter whether oxygen is present or absent, whether it's aerobic or anaerobic. the first pathway is the same. It's the glycolysis. But if there's no oxygen present here in fermentation, that's the only energy-producing step. The product of glycolysis is pyruvate. Pyruvate is pretty toxic to cells, and so cells are going to have a second stage here where they're going to modify that pyruvate, recycle some of the chemicals, and so there's a modification stage. And these are basically the only two stages. And again, we've already talked about glycolysis as a stage, so we're not going to go over that again. It comes from anaerobic respiration, or 2 ATP. So very inefficient. Only about 4% of the energy in glucose actually gets used during anaerobic respiration. And there's a couple of different types of fermentation, and these are certainly utilized commercially on very large scales. The glycolysis is the same for all organisms that generate ATP without any oxygen. But what's different is the modification step, that second stage. And so if we take a look, on the left here is lactic acid fermentation. Many bacteria are anaerobic and they can do this. This is heavily used, for example, in the cheese and the dairy industry. If you take some glucose and feed certain bacteria in a no oxygen environment, they will undergo glycolysis, generate a couple ATPs worth of energy, and those 2 pyruvates as a waste product. Then during the modification, those pyruvates are converted to a chemical called lactate or lactic acid. Again, that's going to be less toxic for the cell. That also recycles those coenzymes then that can be utilized over again. But because there's no oxygen present, there is no citric acid cycle, and there certainly is no oxidative phosphorylation. So all the energy in those lactates and in those NADHs, those coenzymes, are basically unusable for those cells. They're not going to be converted into ATP. Our other type of fermentation might be more familiar to you. That's alcohol fermentation. We see that commercially with yeast, certainly in the brewing industry here in St. Louis, or in the baking industry as well. And glycolysis is the same. We're going to generate a couple ATPs and a couple pyruvates, but then those pyruvates are converted into alcohol, in particular ethanol, which is a 2-carbon alcohol. And again, that's less toxic than pyruvate. It allows the cell to recycle some of those coenzymes, but we're going to see very little energy from that process. So to wrap up or to summarize cell respiration, again, very complex pathway. We're certainly looking at an overview here, and it's all about energy conversion, getting energy converted from potential energy in carbon-carbon bonds and those macromolecules, converted into ATP form that a cell then can use. And aerobic respiration, we said, was with oxygen. That's going to require our 3 steps: glycolysis, citric acid cycle, and oxidative phosphorylation, and that's going to generate about 30 ATP per glucose, which is pretty efficient, about 60% efficient. Again, that's more efficient than most cars, for example, today, converting gasoline into usable energy. If there's no oxygen present, some organisms can undergo anaerobic respiration. Without oxygen, it's very inefficient, only a couple ATPs per sugar, and there's 2 major stages: glycolysis, which is the same as with oxygen, and then that modification stage, and that's where we can see either lactic acid or alcohol fermentation, different stages of that modification.